Okay, thank you, Sid. So it's a pleasure to be here to share some results from my lab, and really this talk is about genetic variation in the end. I'm going to begin by talking a little bit about the importance and origins of genetic variation and its role in understanding quantitative trait variation in trees. I'll then tell you about a functional genomics resource that we've developed to help evaluate a specific type of genetic variation, copy number variation in Populus. And then I'll give you a couple of examples of how we've used this to scan the popular genome for regions controlling poorly understood traits that are important for trees. So I really don't have to stress to this audience the importance of genetic variation. It's certainly the currency of speciation, so ultimately the differences in the shapes of these leaves from different tree species is a reflection of genetic variation. But importantly, genetic variation within species and populations is critical for the adaptive potential of those species and populations. And this is especially important when we're thinking about, for example, how forest and forestry species may respond to climate change. So for forest biology, most traits that are important, not all, there are some simply inherited traits, but most important traits are quantitative. So for example, for commercial forestry, this graph is showing wood-specific gravity, the frequency of individuals with increasing wood-specific gravity within a loblolly pine pedigree. So it shows continuous or quantitative variation, and the same thing would be true if you looked at yield, for example. Additionally, most adaptive and ecologically important traits are also quantitative. So the timing of bud set and bud flush, for example, would also show quantitative trait variation. Now, as Richard just showed, increasingly we have the power to scan genomes and identify genes that are influencing quantitative traits. So for example, now using sequencing, we can actually scan genomes for single nucleotide polymorphisms, and we can look for associations between those SNPs and traits across the genome. Now, one thing that often happens, and this varies depending on species and traits, but even though we have this genome-wide information, we typically cannot predict all of the phenotypic variation. And undoubtedly, there are many different reasons for this. Some of them are probably computational, or how we model the variation. For example, this is a small part of a complex gene network underlying wood formation in poplar. And this diagram really stresses that it's not just the additive effects of individual genes, but probably as, or more importantly, it's how they interact with each other. And this is very difficult to model typically. But what I want to focus on for this talk is that perhaps there are other sources of variation that we're not even considering or measuring in most studies. So structural variation refers to sequence variation, depending on what paper you look at, that is relatively large, so half a kb or a kb or larger. And this can result in gene copy number variation, where individuals will actually contain varied numbers of copies of individual genes. Now, this is not typically surveyed in plants, but recent sequencing studies stress that this is a very common, in fact, a very surprisingly common source of variation. So for example, this work looking at different poplar species and individuals found a very large number of deletions and insertions affecting thousands of genes across these different poplars. So although we don't usually measure this, it is common. So one question is, how might this be affecting quantitative traits? So this sort of variation can have very complex effects on phenotypes, and I'm illustrating that with this cartoon where we have a chromosome from a Populus nigra and a Populus deltoides, so a hybrid. And we're pretending, for example, that this green gene encodes a transcription factor that positively regulates other genes. And this output refers to the number of transcripts being produced. Now, if we change gene dosage or copy number, for example, by increasing the copy number of this green trans regulator, we not only change the expression potentially of that gene, but also of its targets. And because we know that transcription factors often regulate hundreds, if not thousands of other targets, this kind of variation can be quite complex in its output. So in this project, what we wanted to know is what is the relative importance and contribution of this sort of variation to quantitative traits in poplar. So for this experiment, what we did was a controlled cross that's often used for commercial forestry between a Populus deltoides female and a Populus nigra male. But in this case, we took the pollen and we treated it with gamma irradiation to cause chromosomal breaks. Now, the reason that we're doing this is to try to create structural variation. Importantly, this is not transgenic, so we can actually do field trials with these individuals. And you could also use the same approach in commercial hybrids. And one thing that you can do with this sort of population is shown here, where we have pretend phenotypes here, big trees, small trees. And what we can see is for this region of the genome, if we increase dosage, we get a larger tree. If we decrease its dosage, we get smaller trees. So by using a large population of these trees, we can actually march across the genome and look for regions that are affecting traits we're interested in. Now, this is an example of how we karyotyped several hundred individuals from this cross. What you're looking at in this graph are sequencing reads mapped back to the 19 chromosomes of this individual F1. And you can see that for the most part, most chromosomes, the read number is bouncing around the diploid level. But here in chromosome 1, we have additional reads. In this part of chromosome 3, we have fewer reads than expected. And we can use that in combination with SNP information from the parents to infer the karyotype, which for this individual includes an additional portion of chromosome 1, and it has a deletion in this part of chromosome 3. So we've sequenced over 600 genotypes in this way, and we've entered them into field trials. And we have measured an exhausting number of traits, and I'm just going to share a few of these with you today. But I want to point out that the analyses that I'm going to be showing you were done by a very talented postdoc, Eloise Bastians. This is the general statistical approach. If you like, you can just think about the cartoon I just showed you. But what she did, and of course this was for several hundred individuals, but for each individual, she would create a genomic bin based on the breakpoints from additions and deletions across all the different lines. She would then assign a relative gene dosage to each individual for each bin. So for example, for this individual, it has 3 copies of this chromosomal segment within a diploid background. So 3/2, or a gene dosage of 1.5. And by basically doing a statistical association between dosage for each individual within each bin, she was able to look across the genome to look for positive associations between relative dosage and a phenotype. In this case, we're seeing that trees that grow bigger have an additional copy of this bin, and trees that grow smaller have fewer copies. So this is one example of the type of output from these scans. So this is showing spring phenology and fall phenology on the outer rings of the circus plot, and the biomass output of individuals as map QTLs in this inner ring. Here on the outside, these are actually the genomic bins I just described, and the blue is showing you the relative coverage across those bins. So a few general things that we can see for these traits, they behaved a little bit differently. So phenology-related dosage QTL, as we call them, could cumulatively explain over 30% of the total variation. So this is quite a lot of variation that otherwise might have gone overlooked in a traditional QTL or GWAS type experiment. Now for biomass, this is a more complex trait. These QTL tended to have lower magnitude of effect and cumulatively explained only about 11% of the total variation. Now, one other thing we were able to do was to ask questions about the divergence of genes, because the popular genome underwent a relatively recent genome duplication event. And what you see in the middle here is where we've mapped the paralogous regions of the genome. And if you study this long enough, you can find examples of both conserved QTL, but more often we see divergence of regions that are either acquiring new functions or have lost the ancestral function. This is another way that we summarized these data. So these little dice plots are showing you what's happening in bins under different scenarios for phenology traits on the top and biomass on the bottom. This is gene dosage along the x-axis, so deletions, unaffected, and additions. And one thing that you can see is a very large, so 61% and about 50% of the bins for phenology and biomass had no effect, dosage effect on the phenotype, meaning that almost half of the genome for both these traits was dosage sensitive in some way. Now for the QTL that I just showed you, they fall into these categories, and for phenology, we found both direct correlations as well as inverse correlations between dosage and phenology. Biomass was different, however. Here we found positive correlations where additions would increase trait values, but we never found instances where deletions increased the trait value. Now we think that what we're seeing here is not just unmasking deleterious alleles, in part because we see results like this, where both increasing or decreasing dosage affects the trait in the same way. A few things we can conclude from this, though, in general, is that these traits not only react differently, but results like this show us that the genome is held in an overall state of genome balance that's requiring certain dosage relationships that are revealed when we begin to look at these adaptive traits. So we also looked at leaf shape traits. These are important for commercial forestry. These large leaves are typical of many of the most productive poplar hybrids. But also we see wonderful variation. For example, this desert-adapted poplar, Populus euphratica, has this extreme heteroblasty where it makes different leaves in the spring versus the summer that presumably is associated with its adaptation to this extreme environment. Now these are outlines of just some of the actual leaf shapes from individuals in our population. And what you can see is there's quite a lot of variation. And one thing that's kind of striking is that within this population, you can actually find things that look not only like other poplar species, but other things that look like willows, which were in the same family. So it really shows you how much variation this approach was able to create. So this is another QTL map, simply showing you we were able to map QTL for shape that were independent of size, ones that correlated with both shape and size, and then ones that are associated only with the size of the leaf. But what I really want to show you for this, with leaves, we knew when and how to sample for gene expression. So we're beginning to integrate now gene expression data with our phenotypic and genetic data. And once again, I'm using these old dice plots to show you how genes reacted in terms of deletions, no effect, or additions across bins. And about half of the genes that we surveyed that show expression in leaves either did not show a statistically significant change or are showing some sort of dosage compensation. Now, importantly for our study, genes that show a direct correlation between dosage and expression are very good candidates within the regions that we've mapped QTL to actually be genes causing those phenotypes. So among the other things we're doing with this is narrowing down candidate genes, but also we're doing entire chromosome evaluations of gene dosage sensitivity. The last thing I wanted to share with you is an example of how we can use this resource for examining what you might call orphan traits, so traits that are very important for forestry biology, but where we know very little about the genetic underpinnings or the related mechanisms. And I'll just point out that this work is related to a P3 paper, which, look at that, it's got a citation. I'm so excited. But this is work from Daniela Rodriguez, a graduate student in my lab, and she's really interested in how wood anatomy affects water transport in stems. And this is very important because in poplars, the water transporting vessel elements, their diameter is incredibly important for how stems transport water, in part because flow is proportional to the 4th power of the vessel radius. So a very small change in the size of these cells has a very large effect on not only water transport, but also it's highly correlated with cavitation and embolism in response to drought response. Now, this trait is modified during water stress, and that's illustrated here. This is the same poplar genotype under well-watered and water deficit conditions. And you can see just with your eye that under well-watered conditions, you get larger cells that tend to be less clumped, and here you get smaller cells that tend to be more tightly grouped. Very little is known about this trait, though, so Daniela undertook a heroic project where she's been sectioning the stems of a large number of these trees, and this is still work in progress. But she's digitizing all of these images. She's capturing the information about the size, location, and other properties of the water conducting vessels within the cells, and then using those traits as, in genetic analyses. Now, importantly, one thing she's already shown is that these traits show moderate to high heritabilities, meaning that these are not just passive responses to the environment, but they are genetically regulated. And she's even had, with a preliminary result with part of her population, she's been able to already identify specific regions of the genome that seem to have a strong effect on these traits. So in summary, what I've tried to share with you today is that gene copy number and gene dosage are fundamental sources of genetic variation in poplar. Other people have shown that this type of variation is quite common, and we've shown here in this project that it does have significant effects on quantitative traits. We can use this same approach to dissect the genetic architecture and QTL of complex traits, including poorly understood traits like wood anatomy. And one thing I didn't really go into too much is that the results really underlie the idea that during genome evolution, gene dosage is very important, and that overall the genome has to be held in a state of balance. And with that, I'll take any questions.